This is the video animated version of the Radwa and Radwa podcast, where we aim to communicate scientific research to science enthusiasts. We post 4 versions of each episode, in English and Arabic, and in podcast and YouTube video formats. All the links are in the description below. You will also find our references in the description. We are your hosts, Radwa Ewaisha
and Radwa Sharaf,
PhDs, pharmacists, and molecular biologists. In our episode today, we will be discussing RNA. We talked about DNA last time. Radwa, could you please explain what is RNA?
So there are different types of RNA. In today's episode, we will focus on one specific type called mRNA. The M stands for messenger because it delivers a message. Let's see how this works. We know from our previous episode that in every cell inside the nucleus, we have a copy of the DNA. And the copy of this DNA is important because we need to maintain the DNA sequence intact. We have to protect this DNA from any potential damage or changes in that sequence of letters in order to ensure that the cell's function remains in order. And so when the DNA, a specific portion of the DNA is needed in order to make or synthesize a protein, the cell will make a copy of that segment of DNA. That copy is called mRNA, messenger RNA, because that copy will then be exported outside of the nucleus. So it leaves the nucleus, goes outside into the cytoplasm, and can be used to deliver the message from the nucleus that this specific protein needs to be synthesized inside the cell. And then later, once the mRNA is no longer needed, it can be degraded and broken down, and that's okay because we have the original copy protected in the DNA inside the nucleus. And actually, cells can regulate the levels of gene expression and how much each protein is made inside the cell by regulating how quickly or how slowly this message is degraded or broken down. So if we need to synthesize large quantities of the protein, then the message can be maintained for a long period of time in order to allow for the translation of this message into a protein, versus if we only need small quantities of the protein, then the cell can quickly degrade the mRNA message, so we only synthesize a small quantity of protein. So you can think of our DNA as one high-rise building. We don't necessarily need to see every floor at any given point, but when we need to deliver a message outside the nucleus, we need to go to one specific floor. So we will go into that one floor to read the message, see the surroundings, and then exit that floor. So we don't need to visit every single floor when we only need to visit one at any given point to synthesize a protein.
So we can think of the human body as a building for which the construction plan is all in one book that is kept in a safe place. That is our DNA. Every time the construction people need the plan for one floor, they make a copy of that specific chapter and use it, which is our RNA, while DNA is kept in a safe place. So you mentioned last time that DNA is virtually the same in all of our body's cells. Is RNA also the same in all cells?
So we know that it's not the same in all of our cells, but let's see how that works. The DNA is exactly the same in every cell, but the type of cell and its function dictates which parts of the DNA we need to use in order to synthesize proteins. And of course, some proteins are needed by a vast majority of cells. For example, the proteins required for cell division. Most cells need these proteins. And so you will find the message or the mRNA molecules for these proteins present in most cells. But there are other types of proteins that are very cell type specific. For example, the message for insulin, the mRNA for insulin, that message will only be present in the cells that produce insulin in the pancreas. You wouldn't find messages to synthesize proteins in another cell type that wouldn't be synthesizing insulin. Another example is the message for melanin, the pigment that causes and forms our skin color. That message would only be present in the melanocytes, which are cells in the skin that give skin pigmentation. And so experimentally, we can conduct experiments in order to determine the type of cell we have by its RNA content.
So, how do we know the RNA content? Can we also sequence it like we sequence DNA?
We can, yes, we can measure the level of different RNA species and determine the sequence of letters present in that message via sequencing, just like we discussed in our DNA episode. And this is quite important because, one, if we don't know what this message encodes exactly, we can try to predict the function of the protein that's being encoded in this message through identifying the letters that make up its sequence. And two, because in some organisms, like viruses specifically, they don't have DNA, they only have RNA. And so, in order to sequence and identify the virus, we need to be able to sequence the RNA species itself.
So, to summarize, RNA is another nucleic acid that is found in all human cells, but in different quantities and different proportions, depending on the type of cell. It has a sequence of letters, just like DNA, and some viruses only have RNA.
We hope you enjoyed today's episode. Please don't forget to like and subscribe to our podcast and our YouTube channel. You can also find us on Twitter, Instagram, and Facebook. Feel free to reach out with suggestions for future episodes or leave us a comment directly on our YouTube video. See you next time!